How you doing, guys? Welcome to another video. Still on option B, biochemistry. This is volume 2A where we look at proteins. Let's jump to it. So volume 2A, proteins, we look at what are proteins and how are proteins formed. The IB understandings focus on B2, proteins and enzymes. We have to look at 2 amino acids. We need to look at these things called Zwitter ions. We need to be able to explain melting points and solubilities. Then we need to discuss the structure of proteins and enzymes, and then we need to work out reactants and products from condensation reactions of amino acids. Proteins are found in every cell and are fundamental to cell operation. Proteins are polymers and they're made from smaller monomer units called amino acids. 2 of the simpler amino acids are glycine and alanine, which are on the page. An amino acid always has an amine group and a carboxy functional group. The thing that changes between the amino acids is the Z group, one of the groups sticking off the second carbon. That's why they're called a 2 amino acid. Each of those Z groups is different for all of the different amino acids. So all of them will contain the amine, all of them will contain the carboxy group, but their Z group determines the rest of the structure, and that's the one that plays some important chemistry. So for glycine, the Z group is just a hydrogen. For alanine, it's a methyl group. You can get those Z groups from the data book, so make sure you know where they are, make sure you can locate them so you can look them up. All of the amino acids you will need will be found there. You can also find the amino acids in some protein shakes. I'm sure some of you have had some of that. I've never had any. As a result from the acid and basic groups, the amino acids can form what is known as a Zwitter ion. A Zwitter ion is an amino acid that when it's placed in a neutral solution, the acid group will act as an acid and donate a proton, and the basic group, the amine group, will act as a base and accept a proton. So we get an ion that is both positively and negatively charged. That ion will only exist at its isoelectric point. Its isoelectric point is the pH, which can be found on page 31 of the data book. So here we have alanine, and its isoelectric point is a pH of 6. So that means at a pH of 6, that amino acid will both have a negative charge on the carboxy group because it's donated a proton, and a positive charge on the amine group because it's the basic group and it has accepted a proton. The positive is on the nitrogen, the negative is on the oxygen. The Z group remains unchanged. That is known as the Zwitter ion. If we put this substance in an acidic solution has a large amount of H+. The trick here is to remember that in an acidic solution, the H+, the Zwitter ion will be positively charged as well. So the amine group will accept a proton from the acidic solution, having an NH3+, and then the carboxy group, because there's a whole bunch of hydrogen floating around, it will become what we call protonated. So it will simply have a positive charge. So the carboxy group is protonated and the amine group has acted as a base. If we have a basic solution or an alkali solution contains OH-, so the ion will have a negative charge. And the negative charge will be where the acid functional group acts as an acid, and the negative charge is on the oxygen. The amine group, it's a base, so it won't do anything in a basic solution. So what we can do is we can react these Zwitter ions with either a strongly alkaline or a strongly acidic solution to form either the anionic amino acid or the cationic amino acid. So serine can react with a strongly alkaline solution to form an anion, and it's an equilibrium process. So serine is on the right hand side over there, and it has a pH of 5.7 for its isoelectric point. So the Zwitter ion of serine will be the NH3 with the positive at its isoelectric point, and then it will have the negative on the carboxyl group, and its Z group is this CH2OH. Now, if we react that with a strongly basic solution, what's going to happen is the NH3 is going to donate its proton to the OH minus and form H2O liquid. So what we're going to end up with is just a negatively charged anion. The carboxy group will be the site of the negative charge, and we have our water molecule being produced. Now, if we didn't have OH minus, perhaps we had potassium hydroxide. So the potassium ion would come and sit next to the negatively charged oxygen to form the ionic compound. An ionic bond is a lot more soluble than the Zwitter ion. So if we want to increase the solubility of an amino acid, if we can make it into its ion, it's a lot more soluble. We can do the same thing with lysine. Lysine will react with an acid to form a doubly charged cation. Now, how can that occur? Well, lysine actually has two amine groups. So those two amine groups could both accept a proton, act as a base, giving us a doubly charged ion. So here we have our Zwitter ion for lysine. It has the NH3 group, which has accepted a proton from the carboxy group. It's going to be a high pH because it also has another NH2 group as part of its Z chain. And here's the carboxy group with the negatively charged oxygen. If we react that with a strongly acidic solution, for instance HCl, what will happen here is both of the amine groups will become protonated. They'll both accept a proton, but also the carboxy group will form a COOH. It will get its hydrogen back, giving us a doubly charged cation. The solubility of a Zwitter ion increases when the ion can form more hydrogen bonds with water. So if your Z group contains things that can form hydrogen bonds, that's going to increase your solubility. And the melting point of a Zwitter ion increases as the number of charged groups on the ion and the molar mass starts to increase. So the bigger the amino acid, the higher the melting point. The more charge you've got, the higher the melting point. When a molecule contains a carboxy group and an amine group, a condensation reaction can occur to form an amide or peptide link. We get this functional group CONH, and that links the two molecules together and is known as a peptide or amide. So the carboxylic group reacts with the amine group of another amino acid where water is eliminated. So it's a condensation reaction. We get a joining of the two monomers by this amide group and the elimination of water. So we have our plus H2O at the end. The OH from the carboxy group and the H for the amine is what is eliminated. In the simplest case, two amino acids join together to form a dipeptide connected by one peptide link. The reaction is condensation, and for each peptide link formed, one water molecule is produced. So here we have glycine and alanine. The OH of glycine can react with one of the hydrogens of alanine to form our peptide link. So here is our peptide link, our C double bond O, and then it's linked to an NH of the alanine, and then we have alanine's Z group followed by the carboxy group at the end. This could keep on happening. The more monomers we put in, the longer this chain can go on. Don't forget to add your water to balance the chemical equation. I've highlighted the peptide or amide link, C double bond O bond NH. That's the link that joins the two amino acids together. So glycine and alanine, well, they could combine in a different arrangement, giving us two different dipeptides. The first one we looked at over here, which is where glycine reacted with alanine. What about if we flip them over? Alanine reacting with glycine. Are those two things different? Well, yes, in fact they are, and they will function in very different ways. So here we have our alanine with its amine group at the end, and then its Z group, And now its link will be between the carbonyl group, the C double bond O, and the amine group, the NH, to glycine's amine and its Z group and its carboxy group at the end. This is a different dipeptide to the dipeptide before. A polypeptide is formed when 3 or more amino acids join together to form a peptide chain. So we can have a number of amino acids linked together via a peptide link, and they've been circled there. So here we have valine, serine, cytosine, and alanine, all joined by peptide links, and this can continue along to build much bigger chains. Remember that covalent bonds are what connect the amino acids, and they're via peptide links, which are generally strong. It's hard to break those down. If we combine 4 amino acids, how many water molecules would be eliminated in the process? It would be 3. There's always 1 less water molecule eliminated than there are amino acids. So you can remember it's like an N-1 situation. So molecules made from amino acids are often called peptides. When 2 react, we have a dipeptide. When 3 react, it's a tripeptide. And if we get more and more, they're known as a polypeptide. Once we get up to 50 amino acids, we define that as being a protein. Now there's 4 levels of protein structure: primary, secondary, tertiary, and quaternary structures. And we need to know a little bit about those because they determine the function and the shape of a protein. The primary structure is simply the sequence of the amino acids, so the arrangement of them and the dipeptide link between them. So here our sequence of amino acids: histine, leucine, valine, glutamine, histine, and cysteine. That is the primary structure, the arrangement of those amino acids. They're held together by covalent peptide bonds. The secondary structure, well, within the long protein chains, there are regions of the chains that are organized into 2 types of structures. We can have alpha helixes and beta pleated sheets. These are the secondary structure of the proteins, and they're held together by hydrogen bonds between the carbonyl group and the amine group further down the chain. It comes from the lone pair of the oxygen and the N bond H of the amine having an interaction and attraction, a hydrogen bond further down the chain. So they're not next to each other, but they're further down the protein chain. This hydrogen bonding can either form our helix structure or it can form our beta pleated sheets, depending upon the overall structure of the protein. But it's between the carbonyl group of one peptide link to the amine group of another peptide link much further along the chain. The tertiary structure of a protein is described in the way the whole chain folds itself into a 3-dimensional shape, and its shape is very important. The tertiary structure of a protein is held together by the interactions between those side chains, and this can happen in a number of different ways. The side chains could involve ionic interactions, hydrogen bonds, dispersion forces, or disulfide links. And it's all of these interactions that maintain the shape and the way the protein functions. So because we have a number of these Z groups on the chain, they may fold back on each other and have interactions with different parts of the chain. The first type of interaction we could get between the Z groups is, again, hydrogen bonding between the Z groups. That would create an interaction holding them in their shape, holding the protein together. We could have ionic interactions between an NH3 ion and another group such as a carboxy group that's lost its proton. So we can have ionic interactions that are holding the shape of this protein together. We could form what we call covalent crosslinks or disulfide bridges. Disulfide bridges must contain the amino acid cysteine because it's got sulfur, and then those two sulfurs can covalently bond together to form a disulfide link. That link is much stronger than hydrogen bonding or ionic interaction because it's a physical covalent bond. We can also have dispersion forces happening between Z groups that are nonpolar, such as valine and phenylaniline. The dispersion forces come about because of the electrons. The bigger they are, the greater the dispersion forces. The quaternary structure is how the individual polypeptide chains interact with each other, and this gives the protein its overall shape. So collagen is a fibrous protein because the fibers are long and in a chain. Myoglobin is said to be a globular protein because it folds back on itself. You can see here some of the alpha helixes, and there's a few sections of beta pleated sheets in there. The fibrous ones tend to be long-stranded, the globular ones tend to fold back to give it a different shape. But the shape is important for the function. Collagen has a very different function to myoglobin, and it's all got to do with the shape and the interaction of the Z groups that create the shape of the protein. Okay, so volume 2A, some top tips. Make sure you understand the Zwitter ions, and then when we talk about function, it's all about the shape of the proteins. You change the shape, you change the way the protein functions. Thanks for watching, guys. Don't forget, drop a like on the video, subscribe if you're new, and I'll see you